All right. Thank you, everybody, for joining this One World Signal Processing Seminar Series. And yeah, so welcome. So what I should quickly say was an initiative. Well, it's really to bring people together. Either you are signal processing people or related areas in statistics, machine learning, and so forth. We just want to keep everybody connected and excited about our research in our home confinement life. Okay, so this is really a volunteer effort. So if there are ways that we can improve, send us an email. We'll be happy to listen. And also, if you want to get latest updates, I don't know how many of you are just using the Zoom link provided in our email advertisement. Now, but and then you didn't register. Now, if you want to keep watching the later seminar series, would be nicer if you register so that we can send you update information, including the fact that we might be changing the Zoom login information. And also, I would like to really thank my co-organizers, Hoi To Wei and Zhong Hui Zhang. Now, simply speaking, these young people are much more energetic. I just talked to them how about we try that. Older people will say, oh, let's try it maybe a few months later, then we still haven't started to do it. And they said, like, let's do it. And without their help, it's really possible. So I really appreciate their selfless help. All right. All right. So how does it go? Well, very typical, like many of the online seminars you might seen in the past. We'll spend 45 minutes. I try to, and I hope it's really uninterrupted. But of course, if you really have questions, really want to ask it on the spot, well, you can let us know. We'll see how it goes. Okay, and then for questions, you could put it down on the chat box anytime, and then after the presentation, we'll spend like 15 minutes for Q&A section, and I will try to coordinate with the speaker to address the questions as much as we can. All right, so let me introduce today's speaker, Professor Matthew McKay. He's going to talk about vaccine design. He and I know each other for a while, so we know each other pretty well, and he's in fact some of the very outstanding researchers, and we glad we have him in Hong Kong. A very short bio. He got his PhD from University of Sydney, and right now is a professor at Hong Kong University of Science and Technology, and he is co-recipient of Stephen Rice Prize by IEEE Communication Society, Young Alpha Best Award. Sorry, Young Alpha Best Paper Award by IEEE Signal Processing Society, also Research Excellence Award from University of Science and Technology, and also from ComSoft Young Researcher Award, and many others. He has been with MIT and Stanford as a research scientist. Now, before I pass it to Matt, one very notable thing with Matt's CV is that as an established people in signal processing and communication, it is very amazing that he will leave this safe homey Zoom and adventure something that we couldn't even think about, computational biology, which right now has connections to vaccine design, to coronavirus. Anyway, without further ado, I pass it to Matthew, and let's welcome Professor Matthew McKay.
Okay. Thank you so much, Ken and To and Songhui, for organizing this thing. It's a bit unconventional, but this is, I guess, the times now, and it's great that we can have this opportunity to share. And thank you for taking the initiative to do this. So let me share my screen. Okay. Okay. So hopefully you can lay the pointer. Okay. So the screen's okay? No problem? All right, good. So so just to get started. So yeah, so as Ken mentioned, I'm so up until maybe 5 years ago, I was primarily doing signal processing and communications, and but in that time, we started working on this one. Okay, good. So we started progressively moving into this area of what we call computational immunology, where we're trying to use data analysis, statistics, things familiar in signal processing to try to understand the evolution of viruses, and also, with the main aim is to try and come up with ways to help design vaccines for different viruses. And it's just turned out this year that that with this COVID, with COVID coming along, this is really now taking over a lot of what we're doing. So I'm going to try and share with you a few things that we've done in the past couple of years, mainly through 2 case studies. I'm going to mainly talk about our work on hepatitis C, and then the very recent work on COVID-19. Both of these are related to vaccine design problems. I'll say a little bit about HIV, depending on how much time we have. Okay, but my background is in signal processing, and I'm going to try and present this in a way that I hope it's accessible. Hope that it's not too much jargon and so on. Okay, so yeah, so basically there are 2 major things I want to talk about: the Hep C vaccine design problem, and how we can use computational methods to try to help with that. The work on COVID, and if there's time at the end, there's a few projects that I'll just briefly introduce. So just a couple of slides on background. I'm not going to go into too much biology, but basically we all know, I guess, that viruses are they are invading pathogens that. Well, they're pathogens that invade our cells, and they use our own cells to replicate and then sort of go on and infect other cells and so on. There's many different types of viruses, of course. These are just some here. As mentioned today, I'm going to be focusing quite a lot on Hep C, or HCV, we often call it. So just briefly, no matter what virus we have, viruses can be thought of as can be seen as a bunch of proteins, right? So here's an example of a virus particle, and on the inside we might have some genomic RNA for an RNA virus, but on the outside here we got things like spikes on the envelope, which are just sort of proteins that do a job. So here these spikes can often be used to latch onto human cells and infect them. But so viruses, just think of them as a collection of proteins, and then proteins themselves can be thought of as a collection of amino acids, right? So we can visualize them in sort of 3D type of structure like this, or we can describe them by a sequence of amino acids. So here is an example sequence for a given protein of a virus, and so basically you might think of it as having a finite alphabet of size 20. So there are 20 amino acids. So the sequence at any position, you've got one of the 20 amino acids, and they're all listed here. Okay, so so viruses are basically, think of it as a set of proteins, and these proteins can be represented as a sequence of amino acids. Okay, just some very basic biology to start. Okay, so, and also just because I'm going to be talking about vaccines, and I need to just introduce some few basic concepts about the human immune system, or about the adaptive immune system to be specific. So what happens is when a virus comes in and infects us, what it does is it somehow has ways to get into our host cells. And then when it gets into the host cells, it hijacks the replication machinery inside the cell. And then it uses that machinery to create more viruses. And then these viruses go on to infect other cells, and the situation continues. So if we didn't have any way of combating that, we'd be in real trouble. But we have, on the adaptive immune system, we have an extraordinary adaptive immune system with 2 major arms to combat this. The first one is what I call the B cell arm. So we have these sets of cells in our body called B cells, and they have, on their surface, they have these things which are B cell receptors. And these B cell receptors, there's a huge diversity in our body, but these receptors are sort of scanning. They can sort of scan the outside of viruses or parts of the outside of a virus and try to recognize whether things are foreign. Okay. So imagine this B cell, its receptor actually recognize the outside of this virus, what then happens is that B cell gets activated, it proliferates, and it secretes a lot of these receptors in soluble form into the blood. And this is what are called antibodies. So these antibodies then can go on and they can stick to all of these viruses, and then the immune system has ways of then cleaning them up, which I won't go through. Okay, so that's one way of that the immune system tries to sort of negate viral proliferation. And it's fine if viruses are outside of cells, but as this infected cell, these antibodies will do nothing to kill it. It can just keep creating new viruses. So that's where the second arm of the immune system comes into the story. So what happens is in our cells, when we make proteins, and this could be viral proteins, some of those will actually get expressed on the surface of a cell through something called MHC. So imagine this little peptide is part of a virus being made in this cell. So what happens is the second arm of the immune system, which is the T cell arm, these T cells, we have a whole diversity of these as well, which have their own receptors. And some of these receptors can recognize foreign peptides, right? So imagine if this T cell receptor recognizes this foreign peptide, that it's not a human peptide, it's something from, say, a virus. That's a trigger to this immune system that this cell is being infected and is generating viral particles or pathogenic particles. So then what happens is now the T cell, this will proliferate, and there'll be now tons of the same T cells with these receptors, which can then go on and recognize other cells that are infected. And when the T cell recognizes an infected cell, it basically shoots chemicals into it and kills it, right? So T cells are really useful for trying to identify cells that are actually infected already and then killing them. Antibodies will not do that job, okay? So they're kind of the two main arms, T cells and the antibody or B cell arm. And in this talk, I'm mainly going to be talking about antibodies. But the point is, with vaccination, what you try to do is you try to expose the body to the pathogen or some parts of the pathogen prior to natural infection. And what you're really trying to do is you're trying to generate antibodies and T cells ahead of time, which can actually recognize a specific virus or a specific pathogen, right? So the trick is to try and generate the right antibodies and the right T cells, the ones that are effective. Okay. Now, one important point is once the T cells have recognized the virus or the antibodies have recognized the virus, they will tend to, a lot of them may stick around, right? So that's the purpose of vaccination. It's once you expose the body to, or the immune system to the pathogen, you generate antibodies and T cells, and they're called memory, and many can stick around for a long time, and that's really the basis of vaccination, is to try to induce those things. Okay, so we'll sort of talk more about antibodies later, but just generally with vaccination, we have vaccines for many for many different viruses. Two prominent ones worth mentioning are smallpox and polio. So this is an example where vaccines have completely eliminated the virus from the population, and we don't see smallpox anymore. We see polio a little bit, but it's mainly eradicated. It's largely eradicated. So vaccines have had an enormous effect. But for other pathogens, including the COVID-19 virus, we still don't have a vaccine, an effective vaccine. But not just for new viruses, for old ones like hepatitis C or HIV, we still don't have an effective vaccine. SARS-CoV-2 is actually the virus which causes COVID-19 that some of you may already know. So the question that I'm going to try to go through here is, how can we use data analytic tools, modeling, things that are familiar in signal processing, that I'm sure they're familiar to all of you, that to potentially help in guiding the design of vaccines for different viruses. As I said, I mainly focus on hepatitis C today, but I'll mainly focus on hepatitis C and the COVID-19 virus, and a little bit later about HIV and maybe a little bit about polio, depending on time. So that's a brief introduction. So our work on hepatitis C or HCV was to try to identify antibodies that we think might be most effective against the virus. And so for just a brief introduction to Hep C, so this is a huge problem. It infects over 70 million people in the world. It's millions per year still get infected. It's about 20 to 30% of people with Hep C are asymptomatic and they recover kind of quickly, but then the majority of people, if they're untreated, they actually go on to more severe chronic disease, and it becomes a very significant cause of liver cancer, liver transplants, and so on. It's a very bad disease. Now, one of the biggest challenges with Hep C is that it replicates really fast, and it makes a lot of mutations when it does so, and that makes it hard for the immune system to sort of latch onto the virus. It's similar to HIV in that respect. It's a bit differences, but to some extent that the problem is similar. Now with Hep C, unlike with Hep C, unlike HIV, people do recover. HIV, they tend to not clear the virus, but in both cases there are drugs. For Hep C, there are recent drugs which are really actually successful. But there's a few problems. For a lot of countries, they are, they're not on the free medical list, right? They're basically really expensive drugs. And also a lot of people who have this virus don't actually know it. So they can spread it to a lot of people. It's the same, I think it could be a similar situation now, right? So unless you have a vaccine, it's kind of hard to stop that spread of the disease just with drugs. So there's a lot of ongoing efforts to develop a vaccine, but we still don't have a successful one for Hep C yet. Okay, so let me just briefly describe the problem. So as I mentioned at the start, we can think of a sequence of viral involving proteins, and imagine on the surface protein, the surface protein of a Hep C virus has this sequence of amino acids, and imagine that some antibodies might actually latch on to that part of the surface, right? To maybe a part here, let's say, that's encoded here. Or maybe a T cell can latch onto that part, right? It can recognize, let's say, it's this part of the virus. Okay, that's fine. But then what happens is when the virus replicates, it can make a mutation. Let's say it makes an E to a Q, right? And therefore this virus becomes this virus. And this part of the virus becomes this part of the virus, right? And because of that mutation, the antibodies that were working before may not be able to recognize these parts of the virus anymore. Right, so we say that the virus has escaped the immune pressure through mutation. Okay, now, and if this green mutation doesn't compromise the fitness or makes the virus still function, we have a problem, right? Because the virus has escaped. Now, even if the individual mutations themselves, which escape the immune pressure, are deleterious, they kind of, lower the functionality of the virus, right? There can still be another problem. Okay, so imagine that we have this red mutation now, which can escape the antibodies or escape the T cells. Imagine that's deleterious. It sort of makes the virus not function well. What can happen is mutations can occur elsewhere. Here's a blue mutation, which can restore the function. So in this case, the mutation to P facilitates escape from antibodies or T cells, and the blue guy helps to make the virus function. Okay, so the point is that the dynamics involved with antibody or T cell escape are complicated. The mutations work together and collectively actually control fitness. So we have all these network effects, which really, that's where you start to need intelligence. And I think where signal processing starts to be coming into the story. We need to understand these complicated interactions. Okay, this is, of course, just a toy model here, but just to get across the idea. Okay. All right, so now the problem. Okay, so that was still sort of introduction, but the problem that we solved in this work, and this was published last year, was there are known antibodies which are called broadly neutralizing antibodies on the surface of hepatitis C, which target the surface of hepatitis C. And what's happened is people have shown that experiments have shown that people whose immune system generate these antibodies can clear the virus quicker, or fairly, if they generate these antibodies early, they can clear Hep C. On the bright side, but on the negative side, there's also been escape mutations against them, so they don't always work. More than that, the way that people have defined or determined these broadly neutralizing antibodies is they've often tested, they've defined them as broadly neutralizing because they've looked at different sort of subfamilies of Hep C, what we call subtypes. Okay, they're related viruses, and they sort of tested a reference strain in this subtype and this one, and they found that these antibodies might neutralize the reference strains. But in any subtype, there's a lot of viruses, so it doesn't really tell us how well these antibodies neutralize the family of viruses. They've just checked a few specific ones generally, right? So we don't really know how effective they are, right? These antibodies. There's a lot of questions about that. So that's really the question we wanted to answer through computation. How broadly neutralizing, how effective are these antibodies that people have actually are promoting? And which ones do we think are actually the most difficult to escape? So which ones do we think might be the best for a vaccine to induce? Okay. And the way that we're going to do that was to use sequence data for the surface protein called E2 of Hep C. That's the protein that antibodies target, and use modeling and inference. So develop models to try to answer this question. And we're going to bring in not just sequence data, but clinical data and experimental data to try to validate and test the models too. So that's what I'm going to describe. And so the basic idea of our model is that we have, so the virus replicates in the liver, and we're going to try to simulate this replication. So imagine the virus in the liver, there's actually a lot of different strains of the virus. So there's a whole population of viruses. At the moment here, they're just shown to be all the same. And what we're going to do is we're going to sort of simulate how this evolves. And what happens, we're going to use evolutionary modeling, right, which is a stochastic modeling framework. So what we have is, there's a few basic steps. And this is, so the first is the viruses make random mutations. So we incorporate mutations in the model using known mutation rates for Hep C. But then what happens is strains which are actually viral strains which carry a fit mutation. Maybe say this is fit and this is not fit. The fit ones will tend to grow in frequency. They have a selective advantage, right? So we have to have some, in our model, we have to describe which ones are the fit, what are the fit mutants and what are the unfit ones, okay? And that's where the big problem is, because nobody really knows yet or what constitutes a fit virus and what's unfit, right? So we need to have some way of determining that to have any form of meaningful model. So the idea is for a given strain carrying any sort of mutations, we want to define a fitness landscape. That's what we call it, which is basically for any strain, we want to assign a fitness value to it. So if the strain was of length 2, we could think of it in a 3D plot like this, but actually that's very high dimension. So we want to learn a function from sequence to fitness. Okay. Now, one way to do that is to do experiments. Actually see how fast different strains grow, how well they infect in a dish in the lab, for example. And a number of people have done that. There's about, I'm going to say a few things shortly about it, but the problem is you're quite limited to how many strains that you can test. Right? There's a huge number of possible strains, and so far there's on the order of 100 or so that have been tested. That's not enough to build a model. So what we're going to do is we're going to try to learn this fitness landscape from using computation and data. So the data that we actually have is, we have data, this is the genetic sequences, protein sequences of amino acids from, it's roughly 1,300 individuals, Hep C infected individuals. And each sequence of E2 is about 360 letters or amino acids long. Okay, that's the data that we have. Okay, and we're doing this in subtype 1. Okay, so what we want to do is, the basic idea is simple. It's okay, if we see a virus more, it's probably an indication that it's more fit, like a basic Darwinian evolution type of thing, right? If we don't see viruses you know, at least if we do see viruses a lot, you know, ignoring sampling biases and things, which I won't go through at the moment. You know, the basic idea is you see sequences more, it's probably an indication that you see it more because it survives more. Problem is, you can't just look at frequencies of the sequences you see because it's massively undersampled. So what we did is we use a basic unsupervised simple machine learning approach, or totally simple, but a machine learning approach, which is a maximum entropy approach. And basically what we want to do is we look at what we trust in the data. We trust the mutation frequencies perhaps at each position, and maybe the pairwise frequencies that we observe between positions. But higher order statistics and so on are probably don't have enough data to say too much about, right? And the question is, so basically, can we build a model, a probabilistic model, which, you know, which has which agrees with the mutation frequencies and pairwise frequencies, but makes no other assumptions on the data. And that's, so basically the maximum entropy model tries to do that. What it does, I'll mention it here, it looks for the distribution on sequence space, that's on sequences, for which the entropy, which is written here of the distribution, is maximum, but subject to, okay, the frequency is summed to 1, the frequencies, the individual frequencies of mutations at each position should agree with, this is the frequencies in the model, in our data. And the pairwise frequencies of the model should agree with the pairwise frequencies in the data, but no other assumptions. And it turns out that optimization problem, that's the same as, you can pose it in different ways, but it also is equivalent to finding the KL divergence, the parameters of the model, I'll explain in a second, which minimize the KL divergence between the empirical distribution, basically the histogram of the data, and the model of this form here. Okay. Now this form here, some of you might recognize it. It's an exponential model, and some of you might recognize it as an Ising or a Potts model. It's just in physics, it's a common model. And these are H and the J's here are the parameters that one has to learn. Okay, so you have to try and find the H and the J parameters of this model, which gives you a distribution that's closest in KL distance to your empirical distribution in the data. Okay, so this problem, it's actually not it's a terribly hard problem to solve. There's a lot of work done on this in different fields, but the problem is with the protein that we're looking at, it's reasonably long, and it's actually quite variable, which means that the number of parameters here are actually very large, and therefore the dimensionality of this model is very large, and particularly learning these J parameters is complicated. So in order to address this, we have we've proposed an algorithm. The algorithm we proposed within an earlier paper, you know, on HIV the year before, and basically what we did was we come up with we applied a concept called the minimum probability flow algorithm. It's basically an algorithm that has been proposed in physics, and effectively the problem in trying to solve this, even if you use sort of gradient methods or whatever, is that there's a partition function in here, the normalizing constant, that you can't compute easily, right? So it's really helpful if you can actually come up with an approximate solution and then apply gradient methods to try and refine that. So, and that's really what we did. We came up with an approximate solution to then subsequently apply gradient methods. I won't go through the details of this. I'm happy to describe it if anyone is interested, but just briefly, the idea of it is you want to find some sort of Taylor expansion of this, which you can optimize easily. Okay, so you can imagine that this distribution, or for each set of H and J parameters here, you get a distribution. So you have this whole manifold of distributions, right, for different H and J's, and you're looking for the one which has the closest, imagine this is KL distance, to the closest distance to your empirical distribution in the data. So what we did is what the MPA, the minimum probability flow algorithm does is it comes up with some Markov chain formulation, which starts here, and then for a given set of parameters will evolve you to that distribution at equilibrium. And then, so now for every H and J here, every set of parameters, you get a different you effectively get a different Markov chain that evolves you to that point. So then when you have this dynamical formulation, what you do is you take a small time expansion. And when you take a small time expansion, what it actually does is it comes up with a simple expansion for this k-OR distance for small time. I'm sorry, I'm like, I realize this is not a very clean description, very technical, because, but the basic idea is there's a way of expanding this with a Taylor expansion and giving an alternative solution that you can optimize very easily. That gives you a good starting point from which you can then run gradient methods to actually get to the optimal. Okay, so it's sort of a two-step process. But anyway, so when we apply this algorithm, it actually works really fast, and it infers the max. So we inferred the maximum entropy model very well. This is showing the mutant frequencies in the Hep C data, and this is actually the frequencies in the model, right? So they should line up, and they do, at least within some error limits. The model was not designed to reproduce triple mutant frequencies and things like that, but it actually does a very good job at reproducing statistics in the data that it wasn't designed to do. So it is kind of a good generative model as well. But more importantly, and this is just trying to model the prevalence of sequences. But the question is, does it line up with fitness in any way? Right. So what we did was we looked at the predictions of this model. So that basically prevalence is a proxy for fitness, right? The more prevalent, the more fit. That's how we predict it anyway. So what this is showing is 92 different fitness measurements in the lab, different labs, and with a perfect. So this is the actual the predictions by a model, and this is the predictions of the these are measurements in the lab. Now it's not actually fitness; it's inverse. This is a quantity that's inverse, so you would expect it to go down in a line if it was a perfect fit, and it's clearly not a perfect fit. It's actually pretty noisy. But it is correlated to about 0.7 correlation, and it can do a reasonable job at distinguishing what's fit from what's not from what's unfit. Okay, we're certainly not looking for a precise model, but something that can sort of broadly work out what's good and what's bad. So once we had this model, anyway, this was just a fitness model, but once we had this, we developed the evolutionary model. We incorporated it into this evolutionary model, and then we actually said, what if we put antibody pressure at different points? How long would our model take for the virus to escape the antibody pressure? That's really what we wanted to test anyway. Right? The fitness model is just part of this. So we run many generators. So we would repeat. So if we had antibody pressure on this site, what we'd do is we would run the simulation for many generations, and we would wait until the frequency of mutations at the site where the antibody is targeting is beyond 50%, and then we'd say that escape has occurred. So we're trying to test how long does it take for the virus to escape the antibody pressure. And we run many different simulations, assuming there was antibody pressure on each site. And there's 360 sites, and there are we've tried many different founder starting points, many iterations. Actually, we required the Shenzhen supercomputer to do this on 125 nodes, and each one was 24 cores. But in the end, we come up with predictions of how long it would take to escape antibody pressure from Hep C. And what we found is that mutations which are known escape mutations in patients, right, from antibodies, our model predicted that they were very fast. For other mutations, they were relatively slow. So this is sort of some rough clinical validation that our model can sort of, the ones that we did observe in patients, our model predicted will escape happen fast. Okay. There are various other validation that I won't go through here, but other different clinical validation as well, to sort of give confidence that our model is roughly being able to distinguish what escape times from antibodies. So this is a picture of the E2 surface protein, and what it's showing is for every position, this is a predicted antibody escape time. So if an antibody was to target this part, red basically means it's predicted to take a long time to escape. So the antibody would be good. Okay, and similarly, if antibody would target the blue part, for example, we'd think that antibody escape is very fast. Okay, so really the idea from a design perspective is you want to latch on the antibodies to the red parts. Okay, We also tested, so now just the final, this is like the main result. We looked at a bunch of broadly neutralizing antibodies that are known in patients, and also it's exactly known where they bind, what parts of the surface of Hep C they bind to. And we found that there was about 5 antibodies we found which the escape time seems to be particularly long. We think that these are actually the most promising antibodies for a Hep C vaccine design and should be explored more. There are a bunch of antibodies where escape time was actually very quick. And these have been proposed as being broadly neutralizing, but we believe that these are actually less promising because the antibody escape seems to be quite a bit faster. Okay, so that's from that's really to our advice from this particular work. Using these models, we would recommend these particular 5 antibodies for further follow-up with Hep C vaccine design. We also did related work on HIV. This is the corresponding map that I showed for HIV. And this was the fitness versus, the model versus fitness, model versus experimental fitness. And again, you sort of see a reasonable line up here. So I don't have time to go through that work, but there is an analogous type of work that we did for HIV. So the work that I just described was published last year here. And largely it was done, so Ahmed is a postdoc in our team, and Ray was in our team, but now is at UNSW, the medical school. Okay, now to COVID. So I probably need to speed up a little bit. So the COVID work is technically much simpler, but I hope that it's still maybe of interest. So basically, we started working on this about mid-January, and we put up a preprint on the start of Feb. And so the technical stuff, as I said, is pretty easy, as you'll see. But it's what we wanted to do was come up with ways to help guide the vaccine design for this COVID-19 virus, or the SARS-CoV-2 virus. Now we know that this virus, it's well, I don't need to repeat a lot of these numbers. We all know it's been in the news but these are quite staggering and growing very fast, and very different to SARS and MERS. Okay, so in terms of fatality rates, a lot less, but these numbers, infections and deaths, are way bigger. It's become clear, I think, that there is a need for a vaccine, and really we wanted early on to help try to help with giving recommendations for what might be good vaccine target recommendations. So what I mean by that is we wanted to try to identify which parts of the virus to elicit antibody responses against, or T-cell responses against. Okay, we need to, which parts do we think are going to be most effective? Okay, and that's challenging, and particularly before, because we really, it's a new virus. We don't know anything really much about it. We didn't know much about the biology, how it evolves, how the immune system interacts with it. So, how do you start to sort of work out how to design a vaccine? Okay, so the idea that we had was very simple. So basically, well, first of all, the data. So we had at the time sequences, 120 sequences when we started this, which are genetic sequence, full genome sequences from people infected with COVID-19. And so this was the data when we started was about here, and when we published the work, it was about here. Now we're sort of way up here somewhere, above 26,000 or something as of today, the number of sequences we have. So we have sequence data, and we had sequence data even back in January, and that's helpful to some degree. The other thing, but alone it's not that helpful. Well, it may be, but it's the thing that we proposed here was we exploited the fact that the SARS virus, even though it's different to the new virus, it shares some genetic similarity. So the S protein, the spike protein on the surface, is about 76% similar genetically to this new SARS-CoV-2 virus. And for these, for other proteins, it's even higher. For MERS, just by the way, it's quite a bit different. And MERS is the other kind of coronavirus that's infected humans recently. So this similarity is kind of helpful, perhaps. Perhaps we can use SARS to help inform vaccine design for SARS-CoV-2. So that was our idea, was to exploit not just sequence data, but the fact that people had studied SARS for years, and they knew there was a lot of immunological data for SARS. And so people had known, had worked out which parts of SARS to potentially, where humans can elicit a, protective antibody response or a protective T cell response. So there was a lot of work done in trying to develop vaccines for SARS. So a lot of this information was available, and it was in databases. So that was the second part of the second type of information we took. There was these SARS-infected patients, experiments that actually determined that there were parts of SARS, the SARS virus, so different what we call epitopes, or fragments of the SARS virus, which are epitopes, for which antibodies, which antibodies were responding to, or T cells were responding to in some protective way. So the question is, are these parts where there were effective immune responses against SARS, are there any similarity with SARS-CoV-2? And if so, they could. So what we wanted to do is we wanted to search for, among the SARS epitopes, T cell or B cell epitopes, are any of them have a close genetic match in the new virus? And if so, we would recommend these ones as likely places where we can, likely parts of this COVID-19 virus that can induce, or likely induce, an antibody response or maybe a T cell response as protective. So basically that was the idea, and just to summarize, so it was very simple, no complicated math here. We found that of the SARS epitopes, the T cell epitopes, those parts of SARS that can induce a protective T cell response, about a quarter of them were genetically identical, exactly the same in the SARS-CoV-2 virus. Of the B cell epitopes, these are the ones that generate antibodies, about 16% were exactly identical between the two viruses. So we think that we recommended that these parts are parts of, parts which can potentially induce protective immune responses in both viruses, not just in SARS. Okay, and that's very helpful information for actually developing vaccines which get the immune system to recognize parts of a virus. So this is the SARS spike protein, and this is showing where these 20% basically are. Okay, so it's okay, and we also did a population study as well based on immune statistics, but I won't go through that. Okay, now, but that was earlier. So we've actually developed a program, which is because there's a ton of way more data now. It's over 25,000 sequences. So you can take a look at this if you like. It's a web-based server where we provide vaccine target recommendations for B cell epitopes and T cell epitopes and other things. So this work, there's a bioRxiv preprint, and it should appear in Nature Protocols very soon. And basically, some of the features of it are, it's pretty much it's providing the earlier recommendations. It's really updating them based on new data, and it's providing additional flexibility, and in a nice, easy to use form. So we hope this can be a helpful tool for actually guiding vaccine development going forward. What's really interesting to us, and really I think encouraging, is that of the, so if I look at, say, some of the B cell epitopes that are recommended, SARS epitopes recommended by this algorithm, about most of these epitopes, and this is for the surface spike protein, most of them are in a certain subunit of the spike where cross-neutralizing antibodies have been reported to target. But probably more importantly, there are regions of these epitopes that we recommend that have been shown, right, experimentally to be targeted by cross-neutralizing antibodies. So SARS antibodies have been shown to neutralize this new virus, and they actually recognize this part of the COVID-19 virus. Also, in a preclinical trial, there's immune responses have been recorded, protective immune responses have been recorded against some of the other epitopes that have been recommended here. So there is growing evidence that this, using SARS to inform vaccine development for this new virus, can be quite helpful. On the T cells, and also there's in COVID-19 patients, there's recovered patients. They've been targeting regions found by the software too. But even on the T cell side, it's probably even more interesting because there's amongst some of the T cell epitopes we've found, there's been a study recently where they studied 14 COVID-19 patients. Well, in various studies, 9 of the 14, they studied 14 patients, and many of the epitopes here actually showed up in those patients, were being targeted by these patients. And there was one epitope here, which was actually targeted by 10 of the 14 patients. So this seems to be a particularly promising target. Also in another preclinical trial, this one by Inovio in the US, a DNA vaccine have actually found immune responses against this epitope. It's still, not everything is been shown to be effective, but it's been shown to correspond to protective immune responses, but there is a number which appear to. This is ongoing. Okay, so just to summarize, this is the 2 papers I mentioned, if you're interested. This was, so Faraz and Ahmed here at UST, HKUST have been effectively doing a lot of the work here. And for those of you who are interested, I just list a few what I think are very useful reviews of vaccine development for SARS, for COVID-19. Particularly, I would recommend this immunity one. It's really, really good. Just to give you an overview, if you would like to know what the current state of play is in the area. Okay, I'm really running short on time, so I will, let me see, before, all right. Okay, so maybe, Toe, am I okay for another 5 minutes? Is that a?
Yes, I think we started like 10:05, so we can actually end at like 10:55-ish.
Okay, oh, perfect. You've given me 8 minutes.
Yeah, just wind it up.
Thank you. All right. All right. So let me go back. All right. So I just I'll spend then sort of 7 minutes, 8, on just a few of the other related projects that some of them actually connect probably more to signal processing, particularly than the COVID work, but at least from an algorithm perspective. So, you know, one thing that we had done was we had another paper come out this year where we wanted to ask a basic question, again, using these maximum entropy models that I mentioned earlier, but we wanted to ask a basic question. For poliovirus, the vaccine works very well. For HIV, we don't have an effective vaccine. But polio mutates just as fast, or faster even, than HIV. So why is it so easy to get a vaccine for polio compared with HIV? Right? It's not a completely obvious. So people don't. There's no real easy answer to that. So we wanted to try to explore that, and we did in this work. I won't go through all the details here, but basically what we did is we wanted to come up with a fitness model, basically a fitness landscape for polio. So, given polio sequences, can we predict how fit it is, how unfit it is? And so we did that. It wasn't, couldn't just use a max entropy model. We had to do some sort of filtering and other things as well. It's in the paper, but to account for spatial temporal biases and things like that. But in the end, we did come up with a model which was quite predictive. And then we would compare the fitness landscape of the polio virus with that of HIV. And we found that polio, just many more mutations for polio, even though it creates a lot, many of them just kill the virus. Whereas HIV can make a ton of mutations and remain fit. So it kind of seems sensible, right? That it's kind of obvious maybe to some people that it's just HIV can survive mutations better, but it hasn't really been shown systematically. So by using these 2 different models for HIV and Hep C, these fitness models, and comparing them, and comparing the structures of the landscapes and everything, it's all computational. We were able to actually show that polio is somehow more brittle; it's easier to break, right? HIV can make mutations easier. We created a software package for inferring these high-dimensional maximum entropy models. Also, my background was largely in random matrix theory, so I can't help but talk a bit about random matrix theory. And actually, that's what got me into this in the first place, using random matrix theory to analyze sequence data. So again, I won't go through these details, but what we did was for HIV, we wanted to understand which collections of mutations were happening together, which mediate function and structure and so on. So by looking at, so we did, in order to do that, we generated some robust spectral clustering methods. We took the MSA for HIV, this is for HIV and Hep C, looked at the correlation patterns, looked at the top eigenvectors in the correlation matrices, did some eigenvector cleaning, and that's where some random matrix tools were coming from, very familiar in signal processing. Extensions of things that are familiar in signal processing, but it was quite related to things that people have been doing in a lot of different signal processing applications. And in the end, we found collections, this is for example for HIV, collections of mutations which tended to happen together. And it turned out there was quite a block structure that you observe only when you use this type of random matrix cleaning type of tools. If you don't do that, you see just a big mess. And then what we found that these mutations that tended to happen together were always, in a one-to-one correspondence actually associated with different structural or functional domains. So it was really kind of interesting to see that these different parts of, different functions or different structural parts of a virus can actually induce correlation patterns which are quite distinct from others. I mean, it was really quite surprising, but you needed high dimensional inference tools, signal processing tools, I would say, in order to see this. If you just use PCA or something and standard spectral clustering, you don't see it. So, and we've developed a software package to sort of implement this as well, and so we can apply it to different proteins and things. Okay, so I still have 2 minutes according to my clock. So, and the last piece of work, which I'm actually, some work we're really excited about, is looking at genetic time series. So what people have done, if you look at the mutation frequencies in HIV patients or for flu, it's a big mess, right? Things go up and down, and what happens is mutations compete with each other, and it's really complicated to work out what mutations really are helpful for fitness and which ones are just hitchhiking on the background of other mutations and things like that. So we actually developed diffusion-based inference method using something called path integral methods from physics. This is, I better sort of hurry up, but basically what it does is it looks at the mutation frequencies over time and the pairwise mutation frequencies, or the covariance of mutations over time. Does some sort of integrated inverse covariance. So this looks a bit like an MMSE type of thing that would be familiar to people. But in the end, what this would do, it comes up with a way of inferring the fitness effect of individual mutations based on genetic time series. And this was obtained by using diffusion-based inference methods. Okay, so it's a little complicated, but in the end, it could resolve really complicated patterns and really work out, even when things go up and down and so on, what's really seems to be a fitness enhancing mutation, what's neutral and so on. And I think it's the first method, well, it is, to our knowledge, the first method that can actually do this. Most methods actually do, try to solve this problem just by looking individually at trajectories and not looking at the relationships between them, and it misses a lot of these interactions. So we applied that to 14 HIV individuals. So this work, yes, it's still under review, but hopefully it will appear soon. Okay, so just to finish, so I just have to acknowledge, I've mentioned Ahmed and Ray already, and for us, but the work really is part of a broader effort. We in a number of projects, and Arup Chakraborty I've particularly mentioned. He's been my main mentor and our main mentor. He's worked in this area for many years, and I think we've learned immunology from him. And but yeah, there's a collective effort from a lot of people, so I thank them all. And thank you so much to Ken, To, and Sung Hui for organizing this again, and thank you everyone. Sorry for going a bit over time.
Well, thank you, Matt. All right, so yeah, it's about the Q&A section. Okay, anytime if you guys have questions, you could have your video. Sorry, you could have a microphone turned on, but raise your hand first. But first of all, try to coordinate with Matt on addressing some of the questions. Matt, you can also see it, right? I'll start with the first from To. The question is, if you look at the chat room, is there any temporal information from the E2 data that can be exploited since we are dealing with mutation here and there is an evolution model involved?
Any temporal information?
Yeah.
Well, we kind of, I guess it depends on, you know, we're not doing any inference based on, so we don't have genetic sequence data over time. I mean, some are starting to come out, and that's something that relates to the last part. In terms of temporal, I guess it's quite of a broad question. So I'm not quite sure. I mean, if you had knowledge, say, of viral loads over time or the immune responses, because it can adapt over time as well, that type of knowledge, you could potentially incorporate that in. We don't do that. So it's more broad type of predictions, more rough type of predictions. For instance, we know that, I didn't sort of mention this, but there's certain people that that have been found to control and remove, eliminate Hep C, right? So what we did is we looked at the responses of those people. We know what their immune responses were, and we looked at, okay, well, how long would it take the virus to escape those immune responses, and then compared with other people who who didn't recover, who progressed to disease. So it's more of that sort of broader, we're more asking very rough sort of questions about what seems to be most effective and least effective. But you could potentially, if you had more time series information, you could potentially use it, but it really depends on the type of information.
I guess the point there was that you're assuming sort of an equilibrium model when you infer the fitness.
Okay. No, so in terms of inferring the fitness, yeah, that I didn't go through, but we do have a pretty good understanding of that. It's a semi-equilibrium model. That's right. Not for the last part. The last part we're inferring fitness based on trajectories, but the diffusion thing. But the earlier work, yes. What happens with Hep C and HIV is it is kind of at equilibrium. What happens is the different people have very different immune systems. They push the virus in different directions. And what happens is that when they transmit it to somebody else, it reverts. So you get this quasi-equilibrium or this cloud in sequence space. So so people have actually run simulations. Actually, a Roop's team have come up with, explored this in a physics paper where they had models, and they actually very clearly that, at least through simulations, that by inferring, that the inference that you get based on, an evolutionary model of this form, it looks pretty much like you're inferring from an equilibrium model. It's but you need to argue that. So flu, for instance, it doesn't work for precisely this reason, because flu is so directed, whereas HIV and Hep C is sort of not.
Thanks.
I think flu is mutating quite fast, right?
Nowhere near as far. No, not even close to what HIV and Hep C are. Not even close. That's why every year you sort of, it makes it, a few mutations, and therefore you update the strain a little bit, but it's not, I mean, it's the diversity. I think in a previous talk we'd had compared the tree of flu in like a whole year, and HIV in a patient in a short amount of time, and flu was dwarfed. Like the diversity of flu is not, it's there enough to need an updated vaccine, but not, but no, not any as much.
All right. Then we'll go for the second question then from Joachim. All the examples show mutation expressed as substitution in amino acid sequence. I assume you could also have deletion and insertions. If this happened, how does this affect your statistical model? Well, Joachim, you're here, so if you want to follow up for some questions, you always feel free to turn on your microphone.
I think it was well. Yeah. Let's see what else I can follow up.
Yeah, so I mean, you can, I guess there's various ways here. So you can sort of, for insertions and deletion, you can sort of treat them as sort of separate. You can encode them separately as well. So, but you know, it's not a. I don't think there's an exact science about what the best way to handle those is. So there's, you know,
when you define a probability distribution over the sequences, that requires sequences of the same length, right?
Yes. So yeah. So when you align, right. So basically, when we align things, we make sure they're aligned, and therefore at that, you know, you do take into account. You know, you need to basically take into account if there are assertions and insertions and so on, deletions and so on. But it's when you then encode them, right? So how do you handle them? It's similar to so you get gaps, right? And how do you handle gaps?
Right.
So, you know, there's one way to do it is, so we removed sequences with significant amount of gaps. And other ways to which I think we did in this early work, was you can treat it as a separate amino acid, 21st amino acid. So, or other people try to do imputation as well, but it's, yeah, so I think it didn't really make a difference if I recall. I don't recall the exact details of this, but it's, but generally the way that we would handle things like gaps and so on is with a, yeah, I think we added this 21st amino acid. Sorry.
So Joachim, would you like to go for the second question, or you already got a satisfactory answer for that?
No, so it was a question if there are cases which you can use to validate the work using empirical studies, but I think he answered that with the polio and the HIV question in the later presentation.
Yeah, with the, so with the, I think but just if I may just add a brief follow up, I think with the Hep C work and the HIV work, you know, we try to validate in whatever data we have, right? Whatever clinical experimental data, but it's certainly going to be incomplete. And I think that was one of the biggest challenges in working on this field. It's not, you know, earlier we'd write a paper, here's a problem, here's a proof and theorem and so on. It's very clean, right? But here it's, okay, we have a model and how do we know if it's in any way meaningful? So we try to use anything we have, and that's, you know, fitness. We try to use all of the fitness measurements that we could find from different labs as a primary validation. We often use structural contact maps and so on as well. Looking at, you know, we looked at all of the, whatever we could find about protective immune responses, you know, so for patients that had recovered from the virus or not recovered, you know, progressed to disease, what we knew about their immune responses and calibrated that against our, you know, our escape time predictions. So it's, you know, it's, I'd say the work should be taken more of a comparative sense, right, at a rough form. Okay, this is a way to sort of, what we think to classify these antibodies may be good, these ones might not be so good. And the model has been validated, I think, reasonably well at a rough level, being able to make those distinctions. But I wouldn't trust the prediction numbers that we actually, the fitness numbers or the actual escape times that we exactly get, because it's, you know, there are certain parameters and things underlying that. Yeah.
All right, then there comes the third question, which is from me. Now, you know how we signal processing people do research, right? There's a model, okay? Well, you can choose to believe it or not to believe in it, although most people will believe. And then we build formulation and then algorithms. Now, but how about computational biology? I mean, now that could be extremely difficult, right? You first of all have to understand the basic nature of the data you encounter. You make hypotheses even. Yes. You don't even know your hypothesis is correct or not. So would you mind, you can share some of your views on that?
That's, you know, one of the biggest challenges, I think, in getting into this area from signal processing was we had to accept that we don't know everything, right? And, and you know, we don't have enough data always to you know, we can only sometimes say, you know, give partial validation, right? So I think, you know, one thing that I think that I've learned from my collaborators, right, I would say, because you know, it's quite of a different perspective, is that you cannot just propose a model and say, okay, given this model, this is what the model shows, and that's the end of the story. Precisely from what you just said, because the models themselves are going to be kind of more wrong, I would say, than in signal processing, communications, radar, or whatever. They're well established. So you have to work especially hard to actually get any confidence. And the only way that we, there's different levels of that. The first level that I think is absolutely essential is you need to work on real data, and you need to compare any predictions with independent data, whatever that is, experimental data, clinical data, or whatever. And that's what that's a lot. Ahmed has done an amazing job, I would say, in really understanding the biological literature. It's quite diverse, and getting as much information as possible in terms of clinical outcomes and what's known about antibodies. Just all whatever experimental information that we could actually get our hands on. But I think the next level is even more important, and that is then taking your predictions of your model and actually then working with experimental people to actually see if they work, right? Coming up with targeted tests. But we don't have that in what I've described here. But I think at the very least, you need, it's a big challenge, and I think that you can't just treat it as sort of a EE computer science problem. Here's a model that we're going to come up with, and right? You really need, to convince anybody, you need to actually try and show that it's predictive in some sense against independent data.
You also need to make brave hypothesis, and then verify, and then validate it. It could be wrong, right?
Yeah, and sometimes they're really complicated. Sometimes they're sort of easy. Like the polio one was kind of easy. Like we thought, well, it makes sense that polio just makes more mutations that kill itself, right? But to actually come up with models that can prove that, or at least give evidence of that, and to trust that your models are any good, that's where the challenge is. But yeah, it's certainly a fascinating. I really find the area fascinating, but it does require, from a basic research perspective, it is quite kind of different to the things that at least I was used to in more familiar signal processing territory.
All right. So let's see if there are more questions. Are there more questions? You can either type it down now on the chat, or you can open your microphone. All right. Yeah, is it? Yeah, go on, please.
So just I guess personal curiosity. So I sent you a link. So I'm working with a Swedish biotech company that have now produced actual biomedical assays based on the work of Matt. So they actually coated these assays with the targets that were identified in this study. Are you aware of more of these type of events? More jumping on using the results?
So thank you for telling me about that, by the way. I wasn't, we were not aware, but that was nice to see, and particularly that they had, at least on their website, it says that the peptide pools that they developed seem to be, there was some statement about the responses in COVID, recovered COVID-19 patients, if I recall. But yeah, so I'm aware of one other. We haven't actually. These are totally independent. We've not actually ourselves gone to companies to try to push that. But there was another company, there was a paper by researchers at Oxford that came out. It's on bioRxiv, I think, in the last few days. And they also have run some experiments with the T-cell pool. And they mentioned they got it from another company. I can check that company and give you the details if you like. But yeah, I'm only aware of two at the moment that are manufacturing this.
Okay, are there more questions? Now maybe just a joke, okay? This is my final question. When are we going to have the vaccine?
Yes, that's normally it starts with that question. Okay, so I think well, the short answer is that nobody knows, right? But I think there's more. But that's a bit of a cop-out sort of answer. I think that there's more to the story than that. I think it's
I understand that. So that's I said it's really a joke. Yeah.
No, but I think it's an important question, and I think that it's what we are seeing right now, which is really encouraging, is the clinical trials and preclinical trials are being reported, and it's clear that we can get good antibodies against this virus. Right? It's the neutralizing antibodies that people have actually, that are being found in different technologies. So a vaccine is going to be possible to some extent. I'm pretty sure of that. The questions are more detailed questions about which refer to timing. So, how robust are the how robust are the immune responses? Not just antibody responses, but T cell responses as well. How long do they last? So for SARS, antibody responses did not. There was some clinical trials for SARS. There was a couple, at least, that I'm aware of that went through phase 1 and a number of preclinical. And again, they were promising, but there were issues. For instance, the antibodies tended to be often not long lasting, maybe a couple of years, right? If this becomes like a flu virus, that can be a problem for a vaccine, right? Because you need, protection for a long time. In elderly, which is a big, that's what COVID, that's really the most susceptible, I would say, particularly susceptible population. How well the vaccine can actually work in them. But probably the biggest, I think one of the biggest issues is about safety, right? So in the SARS vaccines, there was, they seemed to work pretty well, but also there was some cases where some vaccines could enhance disease, right? They can cause, and in animals, there was some, I think, liver lung damage maybe. There was some cases where they actually enhance disease anyway. There's things which are specific things called antibody-dependent enhancement, where antibodies can actually enhance disease. It's an issue with dengue and other viruses. Something called cytokine storms, which are sort of T-cell related things. So there are safety issues that, I'm not really aware of if they've been, we'll see. I'm not even sure if they're going to be an issue yet with this particular virus. I'm not sure if people are, but there were some issues with SARS, and I think that the safety part needs to be really carefully addressed. And I think that's something that is going to have a direct influence on the timing of the vaccine. I'm not sure if it's really. I think we're going to hopefully have a vaccine, and I don't think there's just one vaccine either. I think there's going to be multiple, and there's so many trials in parallel now that are promising, and if you want to vaccinate the whole world, and each country want to vaccinate their people as well. So it's we'll see what happens, but so that's what I mean. There's a lot of aspects to this.
There's a lot of aspects, human safety in particular.
Yeah, and I think that's something that shouldn't be. It needs to be carefully managed, right?
Exactly. All right then. So if there are no more questions, join me to thank Matt, and also thank you all of you for joining today's section. And I'll look forward to see you next Friday. Thank you.
Let's clap our. I think let's all clap our hands like even.